Good morning and welcome, everyone. My name is Ellie Thiel. I'm a professor of laboratory medicine and pathology here at Mayo Clinic, and I want to thank you for joining today's Future Directions of Laboratory Medicine webinar. We have two presenters today. Both are in the Division of Computational Pathology and AI here at Mayo Clinic, and it's my pleasure to introduce them. We have with us Dr. Christopher Garcia, who is an assistant professor of laboratory medicine and pathology, and we also have with us Deb Novak, who is an instructor in laboratory medicine and pathology, as well as the administrator for the division. And with that, I'll now turn things over to Deb for the presentation. Deb, the floor is yours.
Great. Thanks, Dr. Thiel. Dr. Garcia and I are excited to spend some time with you sharing some of the work that's been done here at the Mayo Clinic. There is no end to the opportunity of what's possible because everything we do in lab medicine and pathology is data. So we're going to give you kind of a brief walkthrough, a brief tour of how we have made the promise of AI a reality in some of the work that we're doing and some of the activities that we're planning to do here at Mayo Clinic in lab medicine. So we are the Mayo Clinic. We have a large laboratory, but really we all know that it doesn't matter if you're a small community laboratory, if you're an academic medical center, if you're a large health system, if you're a small laboratory in a rural community. We're all in this together. We all are facing the same challenges. We have the same pressures and a shared journey. We all have rising test volumes. The demand on healthcare continues to grow, but our resources are pretty static. We aren't really available. We don't have them available, nor do we have the funding to have unlimited resources. Everyone always wants results faster. They want the highest quality results faster from us. We have workforce shortages. Coming out of the pandemic, we've really struggled in healthcare with workforce shortages and burnout, and people just want to work differently. There aren't as many programs turning out professionals and laboratory professionals, and so that's a challenge that we're facing is how do we continue to grow, provide high quality testing with limited resources. We also continue to try to standardize as well. We know that we want to be good stewards with our healthcare dollars. So standardizing with equipment, standardizing with technology, standardizing with workflow and with systems. And so that creates a lot of additional challenges for us in the laboratory practice. And the laboratory practice, as you know, touches all areas of healthcare, interacting with the patient. And so we're very visible, and it's really expected that we're efficient. So again, we share this journey, we share these challenges, and so we can look at opportunities for how we address these together. Today's learning objectives, Dr. Garcia and I are going to touch on these at different points throughout our talk. We really want to talk about the importance of data, what data we have, as well as where we need to digitize data. We all need to understand implementation, and there's additional resources and expertise that's needed to use these tools. So we'll talk a little bit about that. How do we scale and implement? These are new add-ons to our existing workflows, hopefully simplifying things, but we want to make good decisions about where to invest our resources. And then the challenges. We're very highly regulated in lab medicine and pathology, and so we also need to understand what are these new tools bringing to us and what are the challenges they're bringing to our already complex environment. So let's start with the reality check. I think everybody's been aware probably for the last 5 to 8 years that AI is the buzzword, right? AI. A lot of people wonder, what is AI? It's really artificial intelligence, but there's a lot that people throw around as buzzwords. And there's a lot of upstart companies that are bringing great ideas and innovations forward in other industries, as well as trying to apply those to healthcare. But there's really a reality that AI is a tool. It's not magic. It's not something you can implement that's just going to solve all of your problems. And so we really need to understand, how do we put the right process and infrastructure in place, and what skill sets do we need to build up so that we can leverage these tools. They're not going to solve our problems. They're really going to help supplement the work that we do today and hopefully allow us to operate more in the value add activities by being able to automate some of the mundane or routine activities that we do. And so we'll talk about a few examples of those today. The technology is changing quickly. There's different catchphrases, different buzzwords about every 3 months. You know, for a while it's AI. First it was augmented human intelligence, then it was artificial intelligence, then it's large language models, and then generative AI, LLMs, large language models. So there's a lot of buzzwords out there, and so we really need to understand what those tools are and what are the applications. And we're all still learning together. So as I mentioned, you know, our data is important, right? Everything we do in lab medicine and pathology is data, whether it's numerical data that our instruments are generating. We need to understand where is our data and how is our data being stored. We also need to think about what are some digitization opportunities. If we don't digitize some of these pieces of data, we can't leverage tools to work differently. So here's a few examples. When you think of visual inspection, you know, what are some of the things that you're doing today visually that we could potentially digitize and then apply tools to? So are you looking at things under a microscope? Are you looking at your pathology tissue images? Are you looking at your smears? Are you looking at colonies that are growing on a petri dish? What are we doing to assess specimen quality? Are we doing visual inspections for lipemia or icteric conditions or hemolysis? So how do we digitize those so that we can look at the possibility of applying tools? Also look at your manual documentation. Where are you writing things down? Where are you translating data? Where are you moving information from one form to another? If you're capturing it on an instrument and then recording it in your LIS, are you doing handwritten notes to pass information regarding cases and workups to one another? So where are you manually documenting? And what are some physical processes that today are manual for you? Specimen tracking, matching up barcodes. I know some laboratories have barcode comparators where they scan one barcode and then they scan the plate or they scan the slide or the flat to make sure that they're correlating the right patient with the right material. And where are you doing those visual checks? So, thinking about all these digitization opportunities, sorry, that's a mouthful, where are there opportunities for us to leverage that data to then be able to leverage tools? So we really need to look at that whole ecosystem in our workflow and how are things flowing. And Dr. Garcia and I will touch on that a few different times about how important it is that we look at our current state workflow to identify those opportunities. So some of the applications of AI and machine learning today in the clinical laboratory space that you've probably heard about and read about, we'll share some examples of what we're doing in these spaces today. Image analysis. So you've heard a lot about digital pathology. We're not going to go in depth with digital pathology today. That could be a future topic. But image analysis, scanning what is on a slide and digitizing it so that we can unlock the information that's included. We actually are doing, and Dr. Garcia will share as well, what we're doing in the microbiology space with digitization of slides. Workflow optimization, looking at, how do we look at working differently and working smarter and cutting out steps and touches in workflow by leveraging tools. QC and error detection. How can we look for those defects? And I know that's an interest to everybody in lab medicine. How do we look at those pre-analytic defects? How do we look at defects within our process? And how do we detect errors to help us make the best decisions and not send out results that then need to be revised or amended? And then predictive maintenance. Really our future vision is, predicting what the next steps are, what's possible, what the next step in the workflow is. And so looking at those tools are already being applied in that space as well. And then we'll talk more at the end of the talk too about some of the emerging applications, but you hear about large language models, you hear about ChatGPT, and you hear about some of the different applications of LLMs. Right now we're looking at LLMs for how they can help us with interpretive reports, taking a bunch of data and helping us form that interpretive report for the pathologist or the physician to then be able to validate before they're sending it out to the clinician. And then foundation models are being talked about as well, and those are really complicated. Another catchword that you'll hear about is multimodal. Multimodal is really where you're taking data from multiple technologies or modes of data generated from different technologies and applying them together to then be able to come to a conclusion. So think about taking a radiology image, a molecular result, and an anatomic pathology result, and bringing those together for specific disease states. So that area is growing as well. And then agentic AI is one of the latest buzzwords. And really, agentic AI is really, think about chatbots, but it's really kind of chatbots on steroids. So it's really training a chatbot or a bot to have a persona and a role to do certain tasks, and then hooking those bots together for workflow coordination. And again, it's to help do the routine activities that we're doing so that we can have the value add activities that we need our limited expertise to focus on. Dr. Garcia, I'll turn it over to you to talk a little bit about the life cycle.
Yeah, thank you, Deb. So we always feel it's really important to remind and present AI in a life cycle. So a lot of the things that Deb and I will present are in various stages of this life cycle. And a good place to start is when there's an idea. You're looking for a solution, you have a problem. How do you treat this as an opportunity to discover? So these are sometimes really exciting ideas that come up. And like all things at this early stage in the life cycle, not everything progresses all the way through. But some of the AI that you see out there that doesn't have a product out there, that's ideation, maybe early research, early discovery. The next one is really about turning those ideas into a potential solution. So you're gathering those requirements, you're doing the design work. Both of these are really early on. And then at that point, even though it is an equal size of this life cycle circle, this is actually a very large part of the life cycle in the clinical space, which is the development and validation. So there's lots of different phases there, from a proof of concept, where it's, really, does this have an application to a prototype, which is more in a real world environment, but still controlled, and then like a pilot, where you're testing it in a real world environment and seeing, does it solve a solution? So there's a lot of work there. And it's kind of lumped in here, but there's the development and then there's the validation. And then, as we know in the lab, just like other solutions, it's implementation and rollout and training. It's so crucial in AI to think about monitoring and optimization because it's key as we're working together with AI and having it as part of our toolkit to be able to know when it's doing what we're hoping it's doing and when to know when it's not. Another one would be the eventual scaling and evolution of these. So many of these, and I think it's becoming a standard of care or standard of best practice within healthcare, is a phased rollout. You go in one site, you see how well it does, and then you look to scale it up and to make sure that it adapts to the wider area. But just keeping that in mind, we'll probably, I'm going to touch on a couple of applications, and I'll refer to where it may be in the life cycle. So just like Deb had mentioned, digitization is key. We can't use these really sophisticated tools unless the materials that we're using them on are digitized. So just as Deb had mentioned, in pathology, many cases, this can be a whole slide image, or what's commonly referred to as digital pathology. And parasitology in our labs, we do have a lot of digitization going on, and that would be the ova and parasite slides, as well as some bacteriology and some other applications. But also when it goes to pre-analytics, and Deb had mentioned this, imaging tubes for looking for quality issues, whether there's a clot, there's hemolysis, there's lipemic specimens. All of these are things that we're doing with visual analysis. So if we're going to move that to an AI tool or a machine learning tool, there has to be an image that it can apply that to. And then the last one would be documentation. I think we know in the laboratory and in healthcare, documentation is key. There's so much of it that we generate, but how do we make that easy for a machine to be able to consume and to operate on? So things like optical character recognition for reports, requisitions are key. And we can't even do that unless we have that documentation in a digital form, whether it's scanned or an image. So all of these things are going to be a key part of any kind of solution that comes into the lab when it comes to AI and ML. We are presenting this really heavily as computer vision, just because there's so much out there in this space, but there are other ways to digitize other than making an image. And so I think documentation is a good one. Could be receiving lots and lots of messages from other systems. But again, just think first, wow, I can't use AI if things aren't digital. And then again, I just really want to echo, well, we want to echo this key part that process redesign is really important here. When you are using digital tools and AI and ML, you are, in a sense, optimizing things to either scale up or to be more standardized, more efficient. And you don't want to optimize a process that shouldn't be optimized. So process redesign is key. Looking at your current workflows and what's going on in your lab is important. But also identifying your pain points, your bottlenecks, whether that is throughput, quality, whatever it may be. These are key to being able to understand what tools you can use and how you can apply them. But I think we're all learning in the laboratory this next skill, which is really that redesign. How do we use these new tools? How do we use our teams optimally to be able to create a new process that is capitalizing on all of these tools, making us more efficient, improving the quality, and allowing us to scale up? And this is going to be, I think, a really exciting place for us to continue to communicate amongst laboratories, to share best practices, because process redesign with the inclusion of these collaborative intelligences is going to be really huge. And the last bit would be, now that you've redesigned it, you've put this in place, how do you actually implement it and manage it? So this is basic laboratory improvement 101, I know, but it's also key for us to show AI is just a key component of that. It is the overall process that we have to keep in mind. Remember, it's not magic, it's another tool. All right, so we're going to talk about a couple of case studies. Some of these are going to be, well, we'll just go ahead and dive in. One would be workflow transformation. So the first one would be something that I think that we do a lot in our daily work. And I'm going to bring this in more of a review for research. So while it's in the laboratory, we have a lot of research going on, and because of our information systems and how we've worked traditionally in the past, we have to do sequential document review. So if I have a large amount of cases for review, let's say in anatomic pathology, I am going through a very large stack and reading through and looking for patients with a specific condition, cases with a specific condition for research or for quality. And sometimes that can be very manual. Well, in most cases in my career, it has been very manual. And it can be 2, 3, 4 days of reviewing that and getting the information that I need to. And so the delays sit on me because it requires that expertise and requires that clinical eye, and I become a bottleneck to the progression of that work. On the other side, when we're starting to use workflow transformation with AI tools, whether that's digitization of the text, optical character recognition, and in the one that I'm thinking of, it's the application of large language models to be able to contextualize the information and search it more efficiently. We're able to do a whole lot more in this, in looking at large cohorts of studies and to quickly sort and find the right patients that match the research or the quality application. And so instantly, it's no longer one person using one brain and two hands to go through all of this information, but it is using these tools that can look at everything at once and can really help bring the information back to me. So that instead of me parsing through everything individually, it presents it to me faster. So we've been able to do a whole lot more in our research and supporting some of these initiatives with these tools than we could beforehand, because the bottleneck is no longer one set of eyes on a piece of paper at a time. So for our small team that Deb and I lead and work with, we're able to do a whole lot more because these tools really scale us up and amplify our small team. So this is one that Mayo Clinic has presented on and published on quite a bit. And this is another computer vision application, and it's using some AI tools to review parasitology slides. And there's been some really excellent publications. I highly recommend you look them out and see what Bobby Pritt and her lab have done. But the numbers have been the fact that screening time of looking at very high magnification of these slides for parasites has been reduced up to 90%, which is quite significant. And not only has it reduced the screening time for the technicians, but it has also improved the flagging and detection of abnormalities in those slides. So if we're looking for rare cases, this is a wonderful application. And not only has this, improved the working environment for the staff and improved the quality of the detection for the patients, but it's also really improved the satisfaction of the staff. So there is less fatigue from the normal work within this lab, and also an opportunity to focus more on the critical thinking aspects of the work and less of the really menial visual inspection. And the one that I really loved hearing talking and working with this group is they think of AI as a colleague, not as a replacement. Another one that we have right now, a lot of initiatives going on, and they're definitely in the development, which is looking at the pre-analytical quality. So one that we have mentioned a couple of times is being able to look at test tubes for specific assays and making sure that they fit the quality needs for that analyzer downstream. But this is not only for test tubes. You can also think of this, we have projects in digital pathology looking at the scans and detecting whether a scan slide would meet criteria for diagnosis. Is it too blurry? Are there too many folds? Are there things out of focus? Are there processing issues with the slide? There's a lot of opportunities to provide tools to be able to look at quality. But I think all of these come back to the basic problems that we have, which is we're humans. So when we're inspecting things, we can be inconsistent. Another one is because of that inconsistency, and maybe because of our workflows, that identification of a quality issue can be downstream, where it causes a lot of rework and maybe some bottlenecks. And the last one would be because it's visual criteria that we're using to inspect, we can be variable in what we call. Is this hemolysis? Is this a lipemic specimen? Is it borderline? Is it acceptable? Is it not? That can be very subjective as we train each other. And even myself in digital pathology, something that I might have called acceptable one day may be acceptable or unacceptable in another. And so AI does provide some solutions here by being able to standardize these kinds of calls and having much more firm criteria in how we train them on, is it borderline, is it not? And it is definitely much more repeatable in many, many cases. The other thing that can be huge is that it can automatically flag. So we think about it again as a colleague as opposed to a replacement. So these tools can very quickly bring these things to our attention. And as we're using these in the lab, we're learning best practices from other industries such as manufacturing. One of the tools that we've been using was really used heavily in manufacturing. They use it for manufacturing cars and being able to find the defects in like door manufacturing on the line. And so they use that within milliseconds of production. You can flag whether there's an issue or not. And that's what we're looking to do So our goals are to, again, reduce those kinds of quality issues as early as possible. And the benefit is savings not only in our manual review time, but also in catching things before they go too far downstream, so that we can have earlier intervention and less rework. Here's another one that Devin mentioned that really excited about, that our team is working on and developing at this point. But it really is using these wonderful tools, the large language model tools that are out there, to help interpretive reporting. So we have many cases with very complex assays, and there's a lot of data points, and it takes quite a bit of time for our scientists, our physicians to be able to generate a first draft. And large language models are good for some things and not so great at others. But one thing that it is really good at doing, and they tend to get better and better as the models come out, is summarizing information. And that can be huge when you're looking at more than 5 variables. That's the magic number that they talk about in cognition, is 5 plus or minus 2 variables. Humans can do those really well. Once you go beyond that, it's hard for us to balance those in our minds. And so when we have these assays that are 30 plus variables, and you have a tool like an LLM, it can help put those things into context and very quickly generate a first draft. And so what we're finding is that these tools are great not only at saving time, but they're good at standardizing reports. So it's almost like a template, which can be really helpful. And it also is very sensitive at detecting trends, which is very interesting byproduct of the large language models and how they work. The other challenges that we see with large language models are the same challenges that everybody's seeing. The first one would be hallucination risk. So that's the idea of the LLM giving information that isn't within there. So you can tell that would be very bad to have a first draft that has a lot of false information in there. The other one would be PHI privacy concerns. So how do we use large language models in a safe way where we're not exposing any risks to our patients through PHI leakage? We're grateful here at Mayo Clinic that there's been a large investment in that infrastructure, but even then, we have to think about leakage of PHI from one case to another. So that's a key thing that we're learning to work on and sharing our knowledge and our lessons learned, but we're also learning from the wider laboratory community here. And the last one is, how do we integrate these into our workflows? Because our tools are not natively set up to use these. And so as LIS has become more mature, there's opportunities, but also there's so many different ways to be creative and to integrate these into workflows. I think we're going to see, as these become more common and more crucial to providing patient care, standardized ways to do that. But right now, that's not necessarily the case. So as we're working through these, and as you see the field adopting these tools, there's definitely a trust but verify approach. And there's just so much potential here in these tools and in others that I think, as Deb mentioned, these are not going to go away. We're going to hear more and more about these. And the hope is that we not only hear more and more about these, but they become an integral part of making what we do easier and better for patients. So I want to talk about diagnostic improvements. This is one of the major goals that we have, and it's always a challenge. And as we've hinted, there are some regulatory and some other hurdles to go through, but this is one of the major goals, is how can we use these tools to improve diagnosis across the board. And one of those major areas, especially in digital pathology, is focusing on reducing that variability that comes from us pathologists and from the laboratory, and can we provide more consistent interpretations? So even though the Mayo Clinic, I don't think we're using this outside of anything in immunohistochemistry, but if you go to the large pathology floors, there are quite a few AI companies, and most of their solutions are focused on this, whether it is standardizing or reducing variability and calling Gleason scores in prostate cancer or in breast cancer. This is one of those main focuses of the vendor space right now. And the hope is not only does it improve the standardization of these calls across the board, but also it would reduce error rates like missed missed carcinoma or missed, in situ cases. And the other hope is that it would improve access to high quality care. So if you're able to have digitized cases in areas that maybe don't have specialized pathologists or access to specialized pathologists, that these tools do promote better access to that standard of. So there's a lot going on here. And I think we're really excited about many different areas. I mentioned in frozen section, I won't dive into that too much. That's still really early on in development for us here. But one thing that we have seen, and it's not necessarily AI/ML, but it is digital pathology, is access to those subspecialists through digital means is really key. And I know we at the Mayo Clinic are working really hard at trying to make that a reality. But it is key. How do we use the benefit of digital communication to be able to provide access to better? All right, another one that is really important, and there's some very exciting work in this space, is early disease detection. So I think we feel it very much, and Deb mentioned, the trends in healthcare about workforce shortages and burnout, and a lot of that is the care that we provide. I know I'm literally preaching to the choir, is sometimes too late or is in triage mode. And so there's so much potential for early disease detection using AI tools and machine learning tools. And one of those major things is that this early detection is really about pattern recognition. And that's exactly what these tools do very well, and they learn very well. So detecting subtle changes can be hard for us humans, especially when there are so many different variables. But there are some really exciting applications in this space. I don't want to promote one vendor over another, but you can look in this space. There are vendors working at using early disease detection with their tools. Some things could be a little bit outside of the lab space, but touch us like sepsis detection. You can tell that there's been a lot of interest in this in the past. There have been some high-profile failures, but also some wide adoption of some of these tools. But the idea is to not only detect subtle changes, but to be consistent in detecting those, and in a way that makes clinical impact for patients. And that can be really, really key. And all of these tie into quantitative measurements that may be difficult for us to do as humans. So these tools are becoming, not only the concepts becoming widespread and the value being recognized, but they are starting to get adoption in the market. And we'll be seeing these along the way very quickly. Just one stop, I'm going to say, I know it's a little bit outside the lab, but it has been huge in early disease detection. And maybe you've seen this as you've gone in for eye exams, but now image scans of your retina and looking for changes, especially in pre-diabetic patients or diabetic patients, patients with chronic kidney diseases. These are all things that are looking to be recognized earlier than symptomatic. And so retina and different areas for imaging are being key areas for this screening and for a potential pre-symptomatic flagging of specific conditions. So it's always really interesting. Next time you have an eye exam, keep that in mind. They are working on this, and this is becoming more widespread. So just again, not necessarily a word of caution, just trust but verify. There is a lot of people in this space, and as generative AI is making the development of tools easier and easier, we may even see this vendor landscape expand beyond what we have. And the traditional AI vendors, as we've mentioned in digital pathology and in the lab, have really been focusing on imaging. And some of these have shown great potential or great outcomes for patients in small studies. And the regulatory paths are being established. And so we're seeing more things going on. But as we look at the other tools, some of these really exciting tools are at this point still in the development phase and haven't been able to be validated in a very large study or one that is impactful enough for the guidelines to adopt. Also, I feel like I'm very uncontroversial in saying that regulatory uncertainty is definitely there when it comes to these tools. So it's not just the lab space, but these definitely are, right now, we don't know how they're going to be regulated. And so if we are using these tools, we have to be very, not only very cautious, but very diligent and how we make sure that we're being safe for the patients and for the lab, and that we're using tools that are treated just like that, as tools, and not as another brain or as magic. The other things I would say is, as you're getting or exploring these opportunities, pilot programs are essential. Really starting these out as pilot programs, maybe even they're considering them as shadow pilots, running them on the side without any intervention and seeing what value they have as clinical evidence generation could be really key. So there's a lot of great stuff going on in this space. How do you do pilot programs in AI? But these are crucial for large language models and any of these multimodal. I believe this one is still me, right, Deb? Yeah, okay. So personalized, of course, personalized medicine applications. This is really one of those major keys, and I'm probably, everybody's aware of this, but artificial intelligence or machine learning has been used in personalized medicine in the past. There's no way that we could have had genome sequencing without it. But there are beginning to grow more applications in the multimodal personalized medicine applications. As ADCs or antibody drug conjugates gain more marketplace, we have a couple of them out now, but there's hundreds in development in the biopharma pipelines. That biomarker quantification is becoming really key at tying not only how much is on the slide, but what that means for the patient. Tumor analysis. There are some really interesting personalized medicine applications, not only looking at the H&E and IHCs, but merging those together and looking at what that means for patient outcomes. Could help stratify, does a patient get a 6-month regimen versus a 3-month regimen of a specific drug? So there really is a lot of value that's being developed with these applications for patient care to improve outcomes as well as reduce negative consequences. There's so much interest in foundation models because that not only uses a wide variety of data, but also creates a model that could be used for multiple purposes downstream. And this is an exciting place to look at, but it's not necessarily one that is making a huge impact clinically right now. But the promise is there, and this is definitely something to keep an eye on. I know we have many groups here at the Mayo Clinic invested in research in these areas, and I'm assuming that most of you in academic groups are talking about this or investigating. It'll be very interesting to see how these come into the clinical operations of the laboratory. And the last one that I would mention is predictive modeling. I think we're used to this in the lab space a little bit. It's been black box in the past when you look at some of the algorithms with like fibrosis of the liver and integrating multiple analytes, but it is getting more robust. And I think the field is definitely getting more experience with developing these predictive models. And so this is key not only in looking at the specific tissue and analyzing that, but also in what it means for the patient. It's really exciting to see what this is going to do. This is my last slide for a little bit. I'll hand it over to Deb. But this is key. And it's not only building clinician trust, it's going to be building laboratorians' trust. The first one, we've talked about LLM-specific concerns. You can kind of tell I lit up. This is an area I think there's so much excitement going on, but also it's a key area. How do we know when to trust these? How do we know when to use these? How do we know when not to? And it really requires experience. It requires the opportunity to learn and for us to develop our patterns of how we use them. And it is important to know that these things are safe. So these are key areas that I think will be worked on and developed. I know this is something that we're working on. How do we build this? How do we use these in a responsible manner? The other one for trust building is transparency. These tools can be scary, especially when people say, oh, this is going to replace me, or I don't know what it's doing. I don't think anybody in the laboratory should be trusting the tool if they don't feel comfortable knowing what it's doing. And so there's a lot of work for the vendors and for the developers to work on in this space, as well as for our laboratory community. And so we do have a lot of exciting education initiatives that we're working on in the lab, and also collaborating throughout the Mayo Clinic and learning from other institutions. But it's about educating ourselves. How do we know how and when to use these tools and to have those insights? So it goes both sides. Not only do the tools need to provide transparency, but we also have to be educated knowing how to use these tools and how to critically think about using these tools. And then the last one for the traditional concerns is kind of similar, but it's that whole idea of job replacement and black box algorithms. That is scary. Both of those are very scary topics. I think as we learn along the use of these tools, it will be key, but also as we become more educated ourselves about what these tools are and what they aren't, it will help mitigate those big scary questions of what does this do for me? Does it replace me? Does it hurt me? And when do I know when it's working? But overall, building clinician trust is key. Back to you, Dad.
Great, thanks, Dr. Garcia. Some of the messages that we're sharing, we're kind of sharing a few times because we feel they're so important, right? But one of the things that we can't emphasize enough is change management. This is a big change for us. Healthcare, while we are progressive and innovative, sometimes we're the last to adopt cutting edge things because we're a little bit risk averse. And so we've got to find that balance. As Dr. Garcia said, it's kind of that explore, trust, and verify. But from a change management perspective, we just kind of wanted to share some do's and don'ts. Start with willing adopters. Start with building your champions and your champion network within your workspace, within lab medicine and pathology. Celebrate small wins. There's low-hanging fruit. There's no end to the opportunity in lab medicine and pathology. So celebrate small wins. And that could even be research projects that lead to a publication to get you going. Provide extensive training, and I'll talk a little bit more about that in an upcoming slide. And then listen and adapt, because we don't have this all figured out. As I said, it is continually evolving. So listen and adapt to your audience and to the groups that you're working with. We can't force this adoption, or it'll never be embraced. We can't underestimate the amount of training that we need to do, because we didn't receive this training in data science necessarily as part of our undergraduate or graduate studies. And there will be impacts to our workflow, which I know we've shared a little bit about, and we'll emphasize a little bit more upcoming. And we can't overpromise and underdeliver. This isn't the greatest thing since sliced bread, but it is an awesome tool to help us be able to achieve the goals that we set for ourselves. And so we need to look at where does the right application of these tools, and then how do we make sure that we're measuring and monitoring and controlling the use of those tools. So education is big, and this is something we've been working on for the last 4 years here in our Department of Lab Medicine and Pathology, as we've been standing up our work group, really starting with willing learners. So really trying to identify in our laboratories and on our sites, who are those early adopters that are willing to learn more about informatics, data science, how we can leverage our data, how we can look at process improvement. We've been hosting lectures and webinars. We've had grand round speakers come in from outside of healthcare. We've also been trying to build a roundtable kind of cohort where we can bring others from within our practice and within Mayo Clinic to come and share what they're doing, whether it be neurology or cardiology or radiology, to learn about what are best practices and what are some other applications that others are doing. But it really, again, our foundation is data literacy. We really have to understand what is good data, making sure that we have quality data. We have to know the appropriate use of our data, how we access our data diversity, what is the makeup of our data. So we've actually started a data literacy boot camp We had our first cohort this year. It was really fun. So we had our allied health staff partner with one of our lab directors, and we did a data challenge and did a data project, and we just completed a poster session on that first cohort. But a lot of things like, what are the ways we can use Excel? What are the ways we can use BigQuery, which is a tool you could use with the Google Cloud, which is where we have our Mayo internal hosted environment. So we did a lot of great learnings there, and we're going to continue to build on that. But it's important that we insert skill sets too. As I said, we all didn't receive this training in our undergraduate or professional training. So what programs and certificates can we make available to our staff? As well, where do we need to embed data scientists? Where do we need to embed internships? Now this summer, we're excited. We have 15 data science interns in Mayo Clinic in Rochester. And those are really young minds coming from computer science, engineering, data science, that not only are we getting the benefit of their insightful minds, but we're also exposing them to lab medicine and pathology to hopefully be able to grow and support our field as we go forward. As Dr. Garcia said, the regulatory maze is a maze, but there's not really an end to it, even if you can manage the maze, right? Because it's still being built. So, we currently have, software as a medical device with FDA guidance for AI and ML. So we use that as kind of our North Star for what we can or can't do. But we also need to build this out as it pertains directly to lab medicine and pathology. So the College of American Pathologists have developed digital guidelines, and those are continuing to evolve. We encourage all of our colleagues to get involved in those committees, whether it's at ADLM or CAP or UseCap. There's all of our societies and organizations are forming cohorts looking at this related to their science and technologies. Telepathology, if you're doing digital pathology, it's important that you understand what you can and can't do in those rules of engagement as well. So, those are some of the established pathways, but as Dr. Garcia shared, there's some uncharted territory with LLMs, and what are hallucinations and how do you control and manage for those? What are the privacy concerns with PHI, and how can you protect yourself and mitigate risk? So, we need to balance both what we know and what we don't know and try to stay ahead of that to be risk takers, but also be able to mitigate risk, because healthcare is a very risk averse practice. But we're all pioneers in this space, and we can learn from each other. As Dr. Garcia shared, when you're looking at workflow implementation, we just wanted to reemphasize, roll it out slow, have a pre-implementation phase, think about your current state process changes, where these tools can integrate, where it makes sense, do some piloting, limited rollouts, and then deployment with monitoring. So that is really the best way to do that. And as the space continues to evolve, that'll really help support you to see where you need to adjust and as you evolve, especially into clinical translation. One thing I wanted to share with my operations hat on is think about the ROI and the business case. What we've learned is it's very expensive. It's very expensive to set up tools, to hire engineers, to hire data scientists, to have a hosted cloud that you can work in, to have all of your data stored so you can easily assess it, to digitize all of your data. It's very expensive. So think about what you need from an infrastructure, whether it be your own personnel, your own resources, working closely with your IT infrastructure. What are those ongoing storage expenses? How long do you save things? What are your data retention policies? Because you can save stuff forever, but it costs money. One thing I've said a lot over the last few years is the cloud is not free. There's a perception that if you're cloud versus on-prem, that it's free, and it's not. So you really want to think about what's the ROI or the benefit. It's not all about money, but there is a significant investment, and there's not really reimbursement right now. So think about the benefits of reduction in turnaround time, improved quality, fewer errors, ability to have flexibility for remote work. That total cost to ownership is something we really want you to take a look at. And there's some good tools that some of the national organizations, especially with digital pathology, that have put together. to try to help and assess go, no go, or make or buy decisions. But they're still in their infancy, and I think we can all collectively inform those going forward. We want to share a few, failures and lessons, as we've pointed out some potential pitfalls already, but, pilot thoroughly and scale slowly. We can't emphasize that enough. Invest heavily in education, whether that be supplementing your staff or it be adding and providing training opportunities to your staff. Do deep dive reference checks, as we all should, as to be good stewards of our healthcare dollars with any of our vendors, whether we're buying equipment or we're looking at software or we're looking at someone who wants to partner with us. And really do extensive validation. We don't want anyone to repeat our mistakes. We are learning a lot as we go, and our goal is really to share what our learnings through organizations, through talks like these, through publications, and we learn from our colleagues as well. Build your internal champions. So as I said, we really want to have champions that are respected by their peers, technically curious, patient with others, and vocal about benefits. So champion network is going to be really important. And measure success. So again, we talked about, clinical benefits, and Dr. Garcia shared a lot of potential clinical benefits, right? But we need to think of the whole picture. What are the clinical benefits? What are the operational benefits? We don't want to be just creating more work in an already cumbersome workflow, additional steps, additional pieces of software. So think of staff satisfaction, uptime, how we've integrated it. And then we have to look at the bottom line. Are we getting our value out of that financial investment? And so really looking at all of those as success measures together. Dr. Garcia?
Yeah, perfect. I know we're coming close on the hour, so I may be a higher level for these. But there are a lot of exciting technologies on the near term. So the first ones, we've talked about LLMs that could be in production. Lots of industries are using that right now. Foundation models, basic agents. The medium term, we're going to talk about not only the high level, broadly applicable LLMs, but ones that become much more specialized for our field. We'll be seeing that. Autonomous screening, opportunities for collaboration with agents that weren't there in the past. And then long term, they'll be thinking about true multimodal models, ones that can take in radiology, genetics, multiple different lab tests, understanding where they fit in for a whole patient picture. Thinking of that would be huge. And the last one would be supervised automation. So really having independently collaborating or independently thinking but collaborating systems that work. So kind of like an industry 4.0 picture. But the pace of technology is really accelerating. And I think we can see that with where a lot of investment is going, not only in our field, but in others. We've hinted on this, and maybe this is for later, but agentic AI. This is where you have multiple agents that do specialized tasks, but they interact with one another. So it's like having a team that makes decisions and can do very complicated tasks together. These could be from case triage, quality control, scheduling. That's what we're looking on doing this right now. We actually have an agentic experiment that's being published that is looking at the very complex documentation for our AI lifecycle. So this is something where you can really scale up your small team. Other ones could be for monitoring. For any of the routine quality checks that you do in the lab, there's a lot of interest in building agentic teams for that. But key early lessons are, again, have guardrails, build the trust, know what you need to build the trust, and make sure that you have a human team that can understand what they're doing with these tools, knowing when to pull a plug, when to change things. Those are really key. So remember, these things are kind of autonomous, but they should never be unsupervised. We've touched on LLMs a bit and foundation models, so I think we'll leave this here. And I know that there's a question or two that we would like to get to, so let's go to the next slide. But just thinking about the practical steps, just as Deb had mentioned, do your assessments, make sure that you do planning and think of it as a tool. Remember, these can be extremely expensive experiments. In the lab, research is different than the clinical application. So think of it as a tool, think of it as a business case, make sure that you have the champions. And the last bit would be having those discussions of how you put it in place and plan the pilot. So these are good practical steps that we've, I think, learned along the way how to put these in place. Next, please. The last would be building a community. So nobody can do this alone. We're all learning from each other. It's a very sharing community. Make sure that you identify people who are doing this in the field. Be part of those regional user groups. Find the online forums. Find those people who are willing to share their lessons learned. Of course, it'll be safe. People will do that in a way, but that can be really key. And AI is usually just one part of it. So think of it as, having a community for also negotiating with the vendors for what you need and how to adapt it. On the right are some groups that are doing a lot of work in this space, whether it's the CAP, the Digital Pathology Association. If you have a specialty practice area, I guarantee you that they're doing work and having discussions in AI. So getting attached to that group is really key. And the conferences all these days are really having aspects of this, but the Association for Pathology Informatics is very active. So if your group needs a champion in this space or some content there, they are a good group that seems to provide expertise in all of the subspecialty areas of laboratory medicine and pathology. So just become a part of that community of practice. And we talked about let's stay connected. Deb and I are both on LinkedIn. Or, reach out to your Mayo Clinic lab reps if there's more specific questions you have. But we'd love to take the last few minutes for question and answers if that's possible. So Dr. Thiel, back to you.
Wonderful. Well, thank you, Dr. Garcia and Deb. That's a really fantastic presentation on lesson learned and recommendations. So we do have a couple of questions. The first one is, can you expand on the case study? I think this is for you, Dr. Garcia, the workflow and transformation case study. What exactly was improved for pathologists? And can you expand on the sequential document versus parallel document review process?
Sure. We had a specific project that was going to take a lot of time for review. It was a breast cancer review activity looking at multiple stains, if they were positive or negative on these cases. It would have required, we had thousands of cases to look through. It would have required a lot of time. It took me a lot of time at the beginning to look through each individual case to try to understand what was present and what wasn't. And none of the search tools that we had native in the LIS were helping us make that any shorter. And so we had a large language model application for searching through these, structuring the data, and being able to summarize it really quickly. So it's using these tools that instead of me going through one case by one case, using the large language models to look at that huge amount of cases to make those more concise. So there were small tables of ER positive, HER2 negative, whatever it may have been, and then being able to summarize that into groups. And so using these tools to do our work in a smarter manner, that was the parallel documentation, is having multiple LLMs looking at them and then bringing them back to our group for review. So something that would take me multiple nights and was really making me be, the bottleneck of the progress of this project now was able to be done within a matter of hours.
Got it. Thank you. Well, we are at the top of the hour, so I want to thank our speakers again for presenting today, and thank you to all of you for attending.
Thank you for participating. Please click the button below to complete the evaluation and obtain credit.